California, 1981, and the first clues emerged that a new plague was about to strike the human race. Several cases of a rare pneumonia were diagnosed at the same time as an increased incidence of a rare cancer. Other unexplained conditions became more frequent, but there was an underlying common thread: an impaired immune system. This system fights infection from viruses and bacteria, and in 1982, the collection of diseases associated with immune impairment was named AIDS, short for Acquired Immune Deficiency Syndrome. Scientists soon discovered that these virus particles were linked to the development of AIDS. Since then, they've learned a great deal about the virus and can create computer-generated models of its structure. Its elegance belies a potential for devastation. Worldwide, some 10 to 12 million people, including more than 1 million children, have been infected with the AIDS virus. Over a third of the infections have been in women. The AIDS virus, known as the human immunodeficiency virus, or HIV for short, is just a few millionths of a millimeter in size. A cross-section reveals that it consists of an outer protein envelope that surrounds a protein core. The core contains viral genetic material called RNA that enables the virus to replicate when it infects a cell. HIV-infected people who, on medical examination, appear relatively healthy are said to be HIV positive. There are no obvious signs of disease, although infection can be confirmed by laboratory analysis of a blood sample. However, most research suggests that an HIV-infected person eventually develops the diseases that classify someone as suffering from AIDS. Such diseases, which include various forms of cancer, occur on average some 10 years after initial HIV infection, although they may begin much sooner. AIDS patients also suffer from many infectious diseases, a consequence of an impaired immune system that cannot defend the body against bacteria and viruses. Extreme weight loss is also a frequent symptom. Understanding how the virus causes immune system failure and the subsequent development of AIDS is the key to developing effective treatments and, ultimately, a cure. This requires an appreciation of how the body's immune system works. The human body contains a huge network of arteries and veins carrying blood to and from the tissues. The most important component of blood is the red cells that provide tissues with oxygen. Blood also contains immune system cells that attack and destroy infectious bacteria or viruses entering the body. One such cell is a macrophage, often known as a feeding cell. When it encounters an infectious particle, represented here by the triangular symbol, it rapidly engulfs and digests it. The macrophage now presents a piece of the digested invader on its surface. This piece is called an antigen, a specific marker of something that is foreign to the body. Another immune cell in the blood, a T helper cell, now attaches to this antigen and then releases chemical signals into the bloodstream. These signals stimulate T killer cells to hunt down and destroy cells that have already been infected. Meanwhile, agents called B cells that have already attached to the antigen markers of the invaders respond to the signals by rapidly dividing. They start producing Y-shaped molecules called antibodies that bind to invaders and ultimately cause their destruction. T helper cell signals are also sent to immune system centers called lymph nodes. Lymph nodes, shown here in white, are small structures located in the neck, chest and groin. They contain reservoirs of both T killer cells and B cells. The signals from T helper cells trigger them to join their colleagues in battle in the bloodstream and tissues. So, to recap, in addition to calling for help from lymph nodes, The T helper cell triggers B cells to produce antibodies and killer cells to attack cells already infected. The T helper cell is thus the conductor of the immune system. Ironically, it's also a prime target for infection by HIV that somehow evades immune system attempts to destroy it. Around 1984, Professor Angus Dalgleish was one of the scientists who showed that the reason HIV had such a liking for T helper cells was that it attached to a specific surface receptor. A virus envelope protein fits snugly into this receptor. After docking, the viral contents are injected into the T cell, shown here in blue, by a mechanism yet to be explained. Fortunately, most cells in the body don't have such receptors on their surface and are thus not prone to HIV infection. But how does HIV cause AIDS? Initially, it was thought that T helper cell depletion was the answer. We've seen how T helper cells are vitally important as conductors of the immune response and that they're a key target for HIV infection. In HIV-infected people, the number of T helper cells in the blood reduces over many months and years after infection. A very low number of these cells usually coincides with the imminent onset of AIDS. However, many researchers now think that T helper cell depletion, and therefore a weakened immune system, is not the sole reason for AIDS. Scientists at Britain's Clinical Research Centre believe that part of the answer lies in a cell that has only recently attracted attention, the dendritic cell. Dendritic cells are found in the bloodstream and many tissues of the body. The Clinical Research Centre team have shown that when these cells encounter foreign antigens, they take them to the lymph nodes. In the nodes, the dendritic cells trigger an immune response by stimulating the production of both T killer and T helper cells that then stimulate B cells to produce antibodies. The importance of dendritic cells in the immune response led scientists to investigate whether their ability to stimulate T cell production is affected in HIV-infected people. So they looked at what happened when dendritic cells were infected by HIV in the laboratory and mixed with healthy T cells. Antigens in the form of an infectious organism were also added. Under normal circumstances, dendritic cells would present antigens to T cells and trigger their replication. The samples were left upside down to enhance the presentation process. When the samples were later analyzed for the presence of T cells, the scientists discovered that hardly any had been produced. The dendritic cells were unable to stimulate their replication. So, dendritic cells had lost their ability to initiate an immune response. And when dendritic cells from HIV-positive patients were examined by electron microscopy, there was clear evidence of infection. Tiny HIV particles could be seen leaving the cell surface, and dendritic cells from the blood of AIDS patients had an infection rate up to 10 times that of T cells.
Our evidence certainly suggests that early in HIV infection, dendritic cells are reduced in number. They are not functioning correctly. They're not recruiting T cells in the immune response at a time when T cells are still normal and other cells like macrophages are still behaving normally. So we believe this might be a very fundamental and early defect in HIV infection.
Over the last decade, scientists have unraveled how HIV replicates itself inside an infected cell. After the viral protein core and its contents enter the cell, a single strand of viral genetic material called RNA is released. Chemicals now react with it to produce a double-stranded molecule called DNA. This now moves into the nucleus and integrates into one of the many long strands of human DNA that is the chemical blueprint for creating all our body proteins. The hybrid DNA is now also a blueprint for the production of both viral proteins and viral RNA. These now combine to create a new virus particle that emerges from the infected cell. If many particles are created, they eventually kill their host and can go on to infect other healthy cells. So HIV has hijacked a human cell to replicate itself. If we can break some of the links in this chain of replication, then HIV could be defeated. The British pharmaceutical company Wellcome was the first to attack the stage where RNA is converted to DNA. Wellcome scientists chose a chemical called AZT that they showed could inhibit the conversion of viral RNA to viral DNA in laboratory experiments. Initial trials of AZT proved promising. But recent extensive studies of the drug have not shown an increased survival for HIV-positive people, although there may be some benefit for those who've developed AIDS. However, some researchers believe that AZT may prove more effective if combined with other similar drugs that are currently being tested. Another potential replication target is the stage where viral proteins are produced. Here, at the Medical Research Council's Laboratory of Molecular Biology in Cambridge, scientists have been looking at this stage of the replication cycle to discover ways in which it can be interrupted.
Is there any preference for reaction?
Dr. Jonathan Kahn, a biologist, and Dr. Mike Gate, a chemist, have teamed up to investigate how the genes within the viral DNA control the production of new virus particles.
We can then get some low level of incorporation.
The integrated viral DNA actually contains 9 genes, represented here by the 9 colored bands. They code for a range of proteins, most of which eventually combine to create a complete viral particle. Initially, some of the genes are encoded in a single strand of RNA that is copied from the viral DNA. It now moves out of the nucleus and attaches to a protein production factory where it triggers the creation of viral proteins. 2 of these proteins, called TAT and REV, then enter the nucleus and attach to another copy of RNA. This triggers an extension of the single strand to create a copy of all 9 genes. It now moves out of the nucleus and acts as the blueprint for the production of all the viral proteins needed to form a complete viral particle. The Cambridge researchers have discovered the precise points at which TAT and REV interact with RNA to trigger this process. The next step was to investigate how TAT and REV bind to the RNA. So the scientists used genetic engineering techniques to create new strains of bacteria, strains that were capable of producing large amounts of TAT and REV proteins free from the presence of other viral proteins. Once the right strains had been identified, they were grown into huge cultures. TAT and REV proteins can later be extracted from this cloudy soup of bacteria. Meanwhile, fragments of RNA that mimic the binding sites for TAT and REV were artificially created in the laboratory. This was done by using a sophisticated machine that can produce RNA from the basic chemicals that comprise its structure. By mixing the synthetic RNA with TAT and REV proteins isolated from the bacterial cultures, special laboratory techniques can be used to look closely at the nature of how they bind together. This will eventually provide a detailed physical picture of the interaction between TAT and REV proteins and viral RNA, and may soon have far-reaching consequences.
If we can understand how these 2 proteins, TAT and REV, interact with the grooves of their respective RNA targets, it might be possible for pharmaceutical companies to come up with new drugs which will interfere with these interactions. And of course, these interactions only occur in HIV-infected cells and not in normal uninfected cells.
Another potential target within the life cycle is where viral proteins are packaged into a viral particle. This process depends on a specific chemical called a protease enzyme. At Roche laboratories, scientists have been looking for ways to inhibit this enzyme. They began by using genetic engineering techniques to create bacteria that would produce large quantities of the enzyme that could be subsequently extracted and crystallized. X-ray pictures of the crystals were then used by Roche's specialists in computer modeling to create a three-dimensional molecular image of the protease enzyme. A cavity was revealed that experiments showed was the main site of the enzyme's ability to prepare proteins for assembly into a viral particle. Blocking this cavity could be the key to preventing viral particles being created. So molecules were designed that had the potential to fit snugly into the cavity and therefore inhibit the creation of complete virus particles. Hundreds of chemicals were created in the laboratory and tested for their ability to act as an enzyme inhibitor. Ultimately, one was designed that seemed to have the properties that the scientists were looking for. But would it work when added to cells infected with the AIDS virus? A special laboratory was built to do experiments on such cells. This was to ensure total containment of HIV and eliminate any danger of its escape from the laboratory. Samples of blood infected with HIV were mixed with different concentrations of the protease inhibitor and left to incubate. Cells from the cultures were later examined under the electron microscope. In the samples where no protease inhibitor was added, normal viral particles could be seen emerging from the surface of the infected cell. The protein core is clearly defined as the black dot within the particles. At first sight, the same appears true in samples exposed to the inhibitor. But look closely. Their internal structure is significantly different. They now have a donut-like shape with no sign of the protein core. Further experiments showed that the particles had lost their infectious property. Early trials of the protease inhibitor have shown that it can cause a significant increase in T helper cells. In the next few years, larger trials will confirm whether it has a role in delaying the onset of AIDS. Another possible tactic to help the body fight HIV infection is to enhance the immune system response through the use of vaccines. Vaccines contain infectious organisms treated by heat or chemicals to make them harmless. When injected into the blood, they trigger an immune response because they look identical to an infectious invader. A pool of B cells producing antibodies specific for the invader is created, so when a genuine invader is later encountered, the immune system attacks it before it can cause infection. However, this strategy is thought too risky to use with HIV, so another approach to vaccination was investigated. It was in the mid-1980s that Professor Jonathan Weber began looking at whether it might be possible to isolate a part of the virus that could induce an immune response to the whole virus. He created samples of some of the proteins that make up an HIV particle, and he investigated whether the blood of HIV-infected patients contained antibodies specific for each of these proteins. Professor Weber discovered that after infection, patients had a large and persistent antibody response to a protein in the HIV envelope. But in people about to develop AIDS, the antibody response to a core protein called P24 fell away dramatically. If the antibody level against P24 could be raised, perhaps this could prevent the onset of AIDS. This might be done by vaccinating people with P24 to stimulate antibody production. At British Biotechnology in Oxford, a novel approach is being used to create such a vaccine. It takes advantage of an unusual property of a strain of yeast similar to that used to make bread. This is what a normal yeast cell looks like. But the Oxford scientists discovered a strain whose cells looked like this. They contained many tiny virus-like particles. Although they have a protein structure similar to a virus, they had no genetic material within them and were not infectious. It occurred to scientists at British Biotechnology that it may be possible to use this strain of yeast to create virus-like particles containing P24 protein. So they chemically combined the gene that codes for P24 with the yeast gene that codes for the proteins that assemble into virus-like particles. The idea was that the new genetic construct could be put back into yeast to create a combination of both yeast and P24 proteins. If these then assembled into a virus-like particle containing P24 proteins, it would have potential for use as a vaccine. The genetic construct was put back into yeast cells, and initial results were encouraging. Some of them had incorporated the construct. But were these transformed yeast cells now capable of producing virus-like particles? Giant cultures of the different yeast samples were grown in fermenters. The yeast cells were then broken open by blasting them through a small hole. The resulting liquid extract was then passed through a gel filter to purify the sample still further and to separate proteins according to their size. The fractions were carefully separated to collect samples that might contain virus-like particles. The samples were then stained and inserted into slots within a vertical gel that is subjected to an electric current. This separates proteins according to their size. The further they pass down the gel, the smaller they are. The gel can later be photographed, and the dark bands corresponding to the existence of different proteins suggested that virus-like particle proteins had been created. Electron microscopy confirmed the suspicion. But had the genetic construct succeeded in producing particles that contained P24 protein? Antibodies that attached to P24 were added to samples of virus-like particles. Staining techniques highlighted the antibodies as black dots that had bound to their surfaces. This confirmed the presence of P24 protein and meant that a potential vaccine had been created. When solutions of the vaccine were tested in volunteer patients, they were found to be capable of raising the blood level of antibodies specific for P24. An international trial involving hundreds of patients is now underway, and in the next couple of years we'll have a good idea whether the vaccine delays the onset of AIDS. But if it does work, it'll be cheap to produce and simple to administer.
We know in animals this approach can protect against HIV, and studies have already been underway now in humans, and with some indication of success. However, we still don't really know whether in large-scale studies these sorts of approaches will protect people against HIV, and these studies will have to be done in the future, just more in hope than any understanding. So at the moment, we don't have definitely an AIDS vaccine, and in the absence of a cure, we're really still looking at social means to prevent transmission.
We're making more of these things.
In Britain, media campaigns have been used to increase AIDS awareness and to educate people on the need for using condoms to prevent the spread of HIV through sexual intercourse. This is the most common route of HIV transmission from an infected person to someone who does not carry the virus. Another mode of infection is through the sharing of syringes, particularly among drug abusers. Just a tiny speck of blood in a needle used by someone who is HIV positive can contain enough virus to infect the next user. If people believe they're at risk of infection or have already been infected by HIV, they'll need specialist help and advice. At the St. Stephen's Clinic in London, a unit has been set up to handle every aspect of AIDS.
And you've come in today for an HIV test?
I have.
People come here for testing, advice and counselling. If they prove to be HIV positive, it's vital to trace their previous sexual partners so that they too can be tested. Advice and literature can then be provided so that carriers are aware of their potential for infecting others.
Having a look at and that's going to cover some of the issues we've been talking about in the care.
While there are great efforts to find cures for AIDS and to prevent its spread, doctors are also concerned with treating those suffering its symptoms. The most common respiratory infection in AIDS patients in Western countries is a type of pneumonia called PCP. A frequent symptom is a shortness of breath as a result of inflamed lungs. Chest X-rays can confirm the condition. The shadowing across the lungs in an X-ray of a patient with PCP is absent in X-rays of healthy people. Tuberculosis, TB for short, is the most serious and common condition related to AIDS in Africa and is the major cause of death in HIV-infected adults. Treating TB is difficult, expensive, and not always successful. So early testing for HIV and drug treatment to stop TB developing are vitally important strategies. While cures for AIDS are still on the horizon, treating its symptoms remains a priority. As an international authority on the treatment of AIDS, Dr. Brian Gazzard explains.
What is the problem now is there are millions of people worldwide who are HIV infected, who are suffering from diseases which are treatable and indeed curable. These opportunistic infections have a wide variety of treatments which are available for them. These treatments are often extremely inexpensive, very effective, and should be much more widely used. And I think one of the most pressing needs in AIDS is for the international community to get together to make these drugs available on a wide scale so that nobody suffers needlessly from infections which are highly treatable.
During the last decade, there has been progress, albeit slow, in our understanding of HIV and AIDS. So while responsible sexual behavior is currently the key to preventing HIV transmission, potentially effective drugs and vaccines are being tested, and there is improved care for AIDS sufferers. The next decade may see the defeat of a virus that threatens the survival of millions of people throughout the world.